Next, let's talk about epidemiological studies, of which there are three kinds: descriptive, analytical, and experimental. First, let's talk about descriptive epidemiology. Descriptive epidemiology involves the careful tabulation of data concerning a disease. And this includes where the people are, where they live, when they got sick, their age, how long they were sick, etc., etc. Now, this is a very famous map. This map was constructed by John Snow in the 1800s in London during a cholera outbreak. And you can see here we have the pumps, the public pumps that people were using, and deaths from cholera are the brown spots. So what Dr. Snow did is he took a map of the area, tabulated where all of the deaths from cholera had occurred, correlated it to the pumps, and found that the disease was clustered around one specific pump. Dr. John Snow was the first person to use descriptive epidemiology, and this is one of the more famous examples. By the way, he convinced the city leaders to take the handle off the pump, and the cholera went away. In descriptive epidemiological studies, one of the main things that they try to figure out is the index or first case. Sometimes this is impossible because they've died, they've gotten better, they've moved, they don't even remember they were sick, that kind of thing. But in tracking the progress of a disease, it's good to know where it started out. Next, analytical studies. Analytical studies take all the data that is generated by descriptive epidemiological studies and apply more data to it. So, for example, this is a chart that shows various diseases: rabies, Rocky Mountain spotted fever, salmonellosis. And taking rabies as an example, we're looking at the numbers of cases that have happened in all of the states of the United States. So we take all of that data and we take a look to see, is rabies expanding in a certain part of the United States? Is it going down? Do we need to up our advertisements for vaccinating your pets? So on and so forth. Also, in analytical studies, they compare the sick folks to those that are healthy, that are the same age, sex, live in the same area, etc., trying to look for what causes the passage of a disease. They have a tendency to be retrospective. In other words, these are cases that may have been reported a long time ago. We're looking backwards to look at transmission and different things. We used analytical methods to determine that HIV causes AIDS and that it's primarily transferred through sexual contact. We also have experimental epidemiological studies. So we can apply Koch's postulates to a particular disease to determine cause, transmission, and all of these different things. We can only do these types of studies if it is ethical or if we have an animal model. Also, we can use these studies to test a hypothesis resulting from an analytical study, such as the efficacy of a preventative measure or a certain treatment. The example from the book was that in looking at the data from an analytical study, there seemed to be an increased level of heart attacks or buildup of plaque in the arteries, arteriosclerosis, among patients that had chlamydia philia pneumoniae. So what they did is they took, to test the hypothesis that the chlamydia philia influenced the rate of heart attacks, they took one group that was positive for chlamydia philia pneumoniae, and they let them go their way. They just kept track of them and recorded if and when they had heart attacks. Then with the other group, they gave them an antibiotic that was effective against chlamydia philia. Then they looked at the two sets of data, and there was no statistical significance in the rate of heart attacks. So they disproved their hypothesis that resulted from an analytical study. There is a branch of epidemiology that relate specifically to hospital-acquired infections or nosocomial infections. Now, there are three types of nosocomial infections. There are exogenous nosocomial infections. In other words, these are infections that arise from contacting a pathogen once you're in the hospital. So in other words, it came from outside of the patient's body. Then there are endogenous nosocomial infections. These are basically opportunistic pathogens that the patient has brought in, but because they're sick or injured from something else, their immune system is suppressed and they have an opportunistic infection. Iatrogenic infections, which in Latin literally means doctor-caused infections, result from some sort of procedure that has been performed within the hospital. Inserting a catheter, a surgery, an injection, that sort of thing. Now, superinfections really should go underneath endogenous nosocomial infections. This is where the normal microbiota has been disrupted and one organism multiplies, exceeding the numbers that it should have, and causing disease. Yeast infections after receiving antibiotics, Clostridium difficile infections after receiving antibiotics, these are considered superinfections. There are three reasons that patients will get nosocomial infections. The first one is exposure to numerous pathogens present in the healthcare setting. It makes sense. When you're sick, you go to your doctor's office, you go to the hospital, the emergency room. There's all sorts of pathogens hanging out at hospitals. That is the meet and greet area. Then you have patients who are ill. They're sick with something else. They're injured. They have some other problem, and most of the time they have a weakened immune system. Even if you go in healthy and you have a surgery, surgery can depress your immune system. Then connecting those two is the transmission of pathogens among patients and healthcare workers, from staff to visitors to patients, and even from one patient to another by staff moving from one patient to another. Now, we can't do much about pathogens being in the hospital. That's just the nature of a hospital. We can't do anything about the immune systems of patients, but we can do something about healthcare workers transmitting pathogens from patient to patient or from pathogen to staff to patient. We have instituted through the years from hospital epidemiological studies various procedures. 1. All healthcare workers wear gloves when handling patients. That's just good policy. You can take the gloves off. Any pathogens that have gotten on the gloves go into the biohazard. Some patients, the staff wear masks around them, or the patients wear masks if they have a respiratory infection, or if their immune system is so low that they can't fight off infection from normal microbes that they would encounter in the air, and so on and so forth. You get the idea. Lastly, epidemiology and public health. You're all familiar with the county public health departments. You're probably familiar with that we have state public health departments, and we do have a national health department. It's the Centers for Disease Control, the CDC. They work along with some other various government agencies, all of which are involved in gathering data through epidemiological studies to track the incidence and prevalence of disease. Hopefully they can figure out transmission, and they want to be involved in interrupting disease transmission. They sample water to make sure that there's no pathogens in it, that the total microbial count is low. They inspect food to make sure that it's being prepared in a way that it won't pass disease to people. They track contagious diseases so that we can up immunization, warn people about transmission of various diseases, so that, say we have an outbreak of cryptosporidium at a public pool, you can tell people not to go to the public pools if they have diarrhea, and you shut down the affected ones, so on and so forth. Which leads me into public health education. Part of the role of the public health departments is to educate the public about various diseases that are in the area, what's likely coming, and what to do about it. Well, that's it for this topic and for this chapter. here are reminders of what the learning objectives for this topic are.